Hi, everybody here in person and online. It's a pleasure to introduce to you Ben Peter. He is a group leader at the Planck Institute in Leipzig, and he's working on admixture genomics, which is the reason why I invited him, because that's also a topic that interests me a lot. So he's from Switzerland. He was first working with Laurent Escoffier, working on demographic modeling. And then he did his PhD with Rasmus Nielsen and Monty Slatkin at the UC Berkeley. And after a postdoc with John November, he joined the Max Planck Institute in the department of Svante. So he basically helped him to get the Nobel Prize recently.
I don't think that's true.
I mean, some contribution, yeah. Right? Some. So basically he has published a lot on different aspects of population genomics, spatial genomics as well. But of course, he has worked a lot on admixture genomics and developed methods and very well-working methods to detect introgression from Neanderthals to humans. And so his recent success, which is I think what he will talk about, was the family history of Siberian Neanderthals. And we're happy to have you here and looking forward to your talk.
Thank you. Okay, yeah, so let me just move this over a bit. Yeah, so thanks, Martin, for inviting me here and having me here. It's a pleasure to be here. All right, wonderful. Yeah, so as I was saying, so this is really, all this work has been really collaborative and with many people involved. But as my area of expertise in, right, is really sort of on the sort of translating to the genetic data that we get out of these vegetables into biological insights, or sort of to model the connection between the genetics and the biology. And so this is sort of what motivated me to move to Leipzig 5 years ago. And so now, so in some sense I have like this really cool machine where you can get bones in, and this is obviously a very politically like dubious way of defining it, but essentially what you get out, essentially you screen a lot of bones and you get out this really big mess of DNA, right? And so I think sort of what I like doing is sort of to try and untangle this mess and sort of make that we learn something about the history, right? And as many of you will know, sort of the history of our field of ancient DNA has been really quickly growing, right? So the first Neanderthal genome came out in 2010, and if you look at sort of all the ancient genomes, human genomes that were published like 12 years ago now, the map looks like this. Like a few genomes, and then if you go to 2014, like over 4 years, like there have been a few more, but overall not so much has changed. And it's only in the last say 5, 6, 7 years that people sort of started like first looking at population scale data, so between 2014 and 2016, and then they expanded that to like more and more. And so if you look at 2022, and so this is from a data set that David Reich compiled, which I think encompasses most, but probably not all genomes have been published, we can see that now there's a really huge amount of ancient genetic data that we need to interpret. And so this is, I think, to me, in the biggest challenge in the field is like, how do we take this massive amount of data and learn something about human history and our ancestors, which I would argue many of us are particularly interested in, right? And so one of the issues, if you look at these really old genomes, right, so the ones that are older than say 40,000 years, is there's still very few, right? And many of those are also at very pure quality. So like if you sort of work with these in the individuals, or if you're interested in this time period, we have much less data to work with. And so one way we can visualize that is just look at this figure here, or you look at the estimated age of data sets and the count of all these individual data, and the vast majority of them are in this first, say, 10,000 years, so Neolithic or younger. And if you're interested in Paleolithic data or in Neanderthals, then we have much less data to work with, right? And correspondingly, if we look at sort of a history of what we sort of think how different human groups were related to each other, we have sort of this seemingly very simple picture if you look at the early stage. So this is from a review by Nielsen et al. And so what we think is there have been modern humans, Neanderthals and Denisovans, and maybe there's been some change from some other groups. But then if we look at the more recent history, we get a much more complex picture, right? We see like different continental groups, lots of gene flow between Neanderthals and humans, lots of gene flow between different human groups, between continental groups, but also within different European groups, right? And so I think one of the sort of issues that this, I think, or sort of one thing that sort of with sort of my working hypothesis, that this complexity that we see in the present day or more recent history very likely was also present in more ancient populations. It's just that we don't have the data to really understand this yet. And so I think more data and more method required to uncover that, right? And so I'm very interested in Neanderthals, as many of my colleagues in Leipzig are. For Neanderthals, the data record again is very sparse, right? So this is the like full sort of a region of, or sort of full map of all the Neanderthal genomes for which we have nuclear data, right? And so you can see it's about 18 different points, right? And so there's 3 high coverage genomes, one from Vindia Cave in Croatia, one from Czechoslovakia in Russia, and one from Denisovan Cave in Russia. And then there's a few lower coverage genomes from mostly Europe and one from the Caucasus. But overall, we're working with a very small amount of data, right? And then the other group that I very briefly introduced, I'm going to go in a little bit more detail, are these Denisovans, right? Which are a group that, for so far, we only have like very secure data from Denisovan Cave, which is in southern Siberia, and then some more tentative but very likely findings from this one site in Tibet, where a mandible was found. But overall, for Denisovans, the nice thing is we can pretty much put the entire fossil record on one slide, right? So for the data for where we have securely believe that the Denisovans are essentially these 3 tooth fragments and one pinky bone. And so they are named or called Denisovan 2, 8, 3, and 4. And so this is just in the numbers that we have there. But overall, we have very little data, and we sort of want to learn how did these guys interact with each other. And so one reason that we know that they sort of interacted with each other is that what was published a few years ago was an individual that's called Denisovan 11, or people very often call it Denny, where they found that it had a Neanderthal mother and a Denisovan father. So sort of direct evidence that this individual, or the Neanderthals and Denisovans must have been in contact with each other, right? But we have lots of questions about how common was that? Was this a one-off thing? Was this happen multiple times? Or what were the circumstances of that? And that's sort of what I want to do today, sort of to focus on this region here and talk about sort of 2 stories. One, as Martin mentioned, sort of zooming into the structure of one Neanderthal group, but then also one sort of looking at sort of interaction between Neanderthals and Denisovans in this region, right? And so this is what I'm going to start with first. Right, and so to that, I'm going to talk a little bit about the sort of idea about what does gene flow or admixture actually do to an individual's genome. And so the conceptual idea is that if you have, say, a Denisovan mother and Neanderthal father, right, what you have for each chromosome, they have like one pair, or each individual has two copies of the chromosomes. Like here, the Denisovans in yellow, the Neanderthals in blue, and even offspring of a Neanderthal and a Denisovan, what you're going to have is like one copy from each parent, right? And then what will happen is that you'll have recombination that will mix up your genome, and you're going to have a chromosome that looks something like this, that you have some Denisovan ancestry bits, and followed by some Neanderthal ancestry bits, and some further Denisovan ancestry, and so on. And we call these ancestors admixture tracts or introgression tracts. And then say if this individual lives in a Neanderthal population, right, it will sort of get the, say, Neanderthal, fully Neanderthal chromosome from another individual. And if this process goes on, so if the offspring of these folks here live in a Neanderthal population, what we'll get is shorter and shorter fragments, but also more and more of them. And so what people that study admixture are very interested in is sort of figuring out where exactly are these fragments, how long are they, because it tells a lot about both when this admixture happened, how common it was, and whether there were any like functional or biological importance to this gene flow, right? And so one study that many of you know, or one sort of situation that's been well studied, is the gene flow between Neanderthals and modern humans. So these are 2 figures from Fernald Aki and Brownings et al., where they found ones that, if you look at present-day Europeans or Asians, you find Neanderthal ancestry all over the genome. So here in blue, we have the human karyotype. In blue, we have regions where we find Neanderthal ancestry in European, and red, we find Neanderthal ancestry in East Asians, and you can see that you find it almost everywhere, but there's some regions like this one here where you seem to have much less ancestry. And you can also find Denisovan ancestry, as well as Neanderthal ancestry, particularly in East Asians and people from New Guinea. And so what I sort of wanted to do is, sort of take these methods that people used on people that live today and present day data and sort of apply that to data that we have from people that lived many thousands of years ago, right? And so, yeah, so we're running into trouble, right? So there were not many good methods that allowed us to do that. And the first question is why, right? And so, as many of you will know that work with engineering, engineering has sort of its own quirks, right? It's not as straightforward that you just go to Illumina and send them your library and you get your genetic data back, right? So one is that your data tends to be very highly fragmented, right? So because ancient DNA comes from specimens that lived in or were stored in caves or deposited in caves for many millennia, the DNA tends to be very short. And so typically we get fragments that are in the order of like a few hundred or few tens to maybe most 100 base pairs long. Then the second thing that we have is that usually these fragments are chemically modified. So very often you have these C2U deaminations that are sort of a corrected pattern that we see in ancient DNA data. But sort of, I would say probably the biggest issue that we have with ancient DNA is that the vast majority of Neanderthal bones that we recover are very highly contaminated. And so the biggest part of contamination is usually microbial DNA. So these are sort of the first 3 Neanderthals that have been published. So this is the draft Neanderthal genome that was published in 2010. And from the 3 bones that were sequenced there, about 96% of that data was microbial DNA. About 4% range DNA, and there's also maybe 1% or so that was present-day human DNA. And then for the 2 other high-coverage genomes, these were mainly sequenced high coverage because they actually had a lot for ancient DNA purposes of ancient DNA preservation, so about 10 and even 70% for this one specimen here. But for the vast majority of specimen that we recover, actually it looks more like this. About 99% of the DNA we recover is microbial, maybe 1% is present-day humans because people have handled these bones, and there's only a tiny, tiny fraction, if anything, that actually comes from a Neanderthal. And often you have to sort of try to do something with that, is to, instead of looking at these both chromosomes separately, which is sort of the ideal thing to do, right? So there we have like this one sort of admixed chromosome and this one non-admixed chromosome. We just call a diploid tract. We just say, we say either we have some regions where we have both Neanderthal and Denisovan ancestry in this case, so both colors, then some regions we only have one color, and so on. So we don't try to phase or distinguish the 2 chromosomes that we have in the genome. And this makes the problem but easier, but makes it less powerful, right? So we have less power to distinguish different ancestries. Right. But sort of the whole idea, I think, is that we need better data models to actually use this very scarce Neanderthal data more efficiently. Right. And so this is just a really brief overview of our models. It's a technical bit. So basically what you're interested in is what's called the local ancestry, or which part of your genome are Neanderthal or which ones are Denisovans. And then we have this graphical model here that we have given a local ancestry, we can ask for any particular SNP, what is the genotype, right? So if we know something about Neanderthals and Denisovans in a particular location, we can say, given that you are Neanderthal in a particular region, you can sort of predict what your genotype might be. And then if you sort of assume we know something about how the data is generated and the deamination processes and the contamination processes, we kind of pretend we can have a model that pretends what sequencing data we might get at this particular location, given the genotype. Right? And what we do with that in practice, we use what's called a hidden Markov model. So we just subdivide the genome into tiny, tiny bits and assign each of them a local ancestry. And so I'm going to use throughout this talk this sort of 3-letter abbreviation where I use DN for Denisovan ancestry and NEA or blue for Neanderthal ancestry. And so what we would have here, right, is sort of these sort of sequence on the genome where we have here Neanderthal ancestry fragment, then back to the Denisovan ancestry, and then on the other chromosome, we have Neanderthal ancestry here. But as I said, what we hope to get with my method is to just say that in these two regions here, we have both ancestries. In the first one, we have only the Denisovan ancestry, and here we only have Neanderthal ancestry. But we don't distinguish this case here from this case here, where the first one is Neanderthal ancestry here, the second one, we both would call them the same ancestry. And yeah, so the nice thing about sort of this setup, a modeling setup, is that we sort of can make minimal assumptions and we can estimate pretty much all parameters from the data. So we don't have to say that we know, for example, how diverged Neanderthals and Denisovans are, because we don't have very good estimates for that. Right, then how do we get the genotypes? Again, here I'm just going to very briefly sketch the math for the people that are more technically inclined. But basically, the idea is we use what's called the Baldwin-Nichols model because it allows us to take uncertainty into account. So we assume that we have some Neanderthals and some Denisovans, but we know that Neanderthals and Denisovans that we sequence are probably only not very distantly related to the ones that actually were involved in the gene flow, and we have probably some substructure between Neanderthals, some substructure in Denisovans, and there's been a lot of genetic drift since that gene flow happened, and we can take that into account in, I think I'm going to skip over the math, but essentially the long story short is like if you make this very simple model, you can actually get closed form solutions for the ancestry. So the idea is that you have homozygous ancestry for a particular source. So if you know that both of your copies are in your individual of interest are say either Neanderthal or Denisovans. So what matters essentially is whether these two have a common ancestor before gene flow, and then what matters is how much has the allele frequency changed from your sampling time to when gene flow happened. And so we have two parameters called F and tau that sort of measure. F measures what's probability of coalescence here, so measures sort of the population history in the target, and tau measures the population history in the source. And if you make this very simple modeling assumption, you can actually get these very simple closed form solutions for the probability that you have, say, homozygous or heterozygous genotype given this homozygous ancestry. And then for heterozygous ancestry, so if you have both Denisovan and Neanderthal ancestry, it gets even simpler because all that matters is what you observe from your source sample. So how many, say, ancestral derived alleles you observe at any particular site. And then the last thing is that we worry about what's the probability of having particular sequencing reads that we observe from the sequencer given the genotypes. And here, we assume we have contamination, and we know that contamination is not uniformly distributed in all sequencing data. So, but for example, we know that longer fragments tend to be more likely to be of contaminant origin, simply because ancient DNA will be breaking down, and so it tends to be very short. Versus if you have some contamination from, say, the people working in the lab, that tends to be more younger DNA, so it tends to be longer. And then we know that if you have these damage patterns, it's much more likely to be endogenous DNA, so it's usually less contaminated. And then we also have different ways to prepare the sequencing libraries. So for example, Matthias Meyer or people in the wet lab have used protocols that involve bleaching of DNA data. So they treat the data in a way that sort of preferentially destroys contaminant DNA, and that's also often used. So what we have in some sense is that we have variation between these different reads, right, based on these covariates. And what this means is that sort of this gives us power to estimate contamination for these different parts, right? Because whenever you have sort of systematic differences between reads that have, say, deaminations or not, then we know this must be due to contamination. It cannot be due to any endogenous factors, right? And so what this allows us is to just estimate the contamination for each group of fragments separately. Right, and so to sort of show how this works, I'm gonna use show some sort of proof of concept and then some analysis. And so I use Africans, sub-Saharan Africans as sort of both potentially modern human source or contaminant, and then the 3 high coverage Neanderthals as a Neanderthal source and the high coverage Denisovan genome that we have as a Denisovan source. And so the analysis are restricted to sites that are variable between dark K hominins that we know. Right, and so the way I like to display this result is something like this. So this is for, I don't know if you can get rid of this bar somehow, but this is a specimen called Usti Shim. It's, can we get rid of this? Or can we? I don't know, I don't have a good spot for it. Yeah, yeah. Sorry, this seems important that you. Wonderful. Yes. So now you can see the full. You can see the full thing, right? So this is a specimen called Denisovan. It's a modern human, and what I plot here is how we interpret the genome or the karyotype of this individual, right? So here we have all the chromosomes. On the y-axis, we have the probability of a local ancestry, and in colors, we have in gray modern human ancestry, in blue ancestry where it's modern humans and Neanderthal heterozygous, and in orange where it's modern humans and Denisovan heterozygous ancestry. And you can see that we have these clear tracts here, for example, here on chromosome 21, where we have some Neanderthal ancestry in this genome. And then, but most of the genome, because it's an early modern human, are in gray, so we don't have any evidence of Neanderthal ancestry in this particular individuals. So this is a very high coverage genome. So it's roughly 40x. So each base is roughly covered 40 times, which is a lot. And so one thing you can ask with this method, what happens if you have less data, right? So we start with this 40x genome, and here just showing chromosome 2 to just make things simpler. And well, first thing we do is to compare to other people's methods. So one by Xiaomei Fu that was done in sort of the original analysis of this paper, and then one by Lauritz Skoff that also developed a method for the same purpose, tailored for these high coverage genomes. And we can see that we mostly get very corresponding data, right? So the Fu method in particular just has a lot more noise, but the Skoff method is roughly corresponding to what we find. But then what we can do on these other methods cannot do is we can sort of downsample. And so one thing I did is just tossed out 99% of the data, right? It's just said like, well, instead of a 40x genome, we just have a 0.4x genome, which is sort of, yeah, maybe the average coverage one gets for an ancient genome. And the nice thing about my method is that it affects actually our results very, very little, right? So overall, we get mostly the same sort of pattern, particularly for the long fragments, which are maybe the more interesting ones. We also detect them from much, much less data. We can go even further down to 0.1x and 0.001x, and sort of find that for 0.1x, it sort of works still reasonably well, but then for if you only have one in every 100 spaces covered, then we start losing most of our power. We don't get much anymore. But still, it's a method that works well if you have much less than 1x coverage data. You can also ask, what if you add contamination? So here I just added contamination between 80% or 5% and 80% contamination. You can see the pattern stays mostly consistent. And we can also ask, does it matter which human reference do we use? So we use sort of Africans, use Europeans or East Asians. And again, we find that the patterns don't change so much. So it doesn't seem to matter too much which human group we use. And this is mainly because Neanderthals are quite distinct from all present day populations. Now, what I'm really interested in this sort of study is looking at the Siberian Neanderthals, right? And so here is sort of a timeline, and I think I have not updated it too recently, but essentially this is sort of the sort of a rough sketch of the data specimen from which we have data from. So we have 2 high coverage Neanderthals, the Alta Neanderthal, which is also called Denisovan 5, but I'm just going to call it Alta Neanderthal because it's sort of more common in genetics literature, which is 38x genome. Then Jagirska 8 is another Neanderthal that's 26x, and both of those have very little contamination. And then Denisovan 3 is another relatively young Neanderthal. That's at high coverage. And then all the other specimens that we have, for example, Denisovan 2 is much older and it's very highly contaminated. And then also Denisovan 8, which we're going to look at, is we have only low coverage data and probably quite a lot of contamination. And so sort of the first sort of application is this Denisovan 2. It's this very old Denisovan, it's the oldest Denisovans that we have. And the dating, I think, is relatively uncertain, but it's roughly 160,000 years old. Katarina will correct me, but I think that's sort of roughly so here I have the confidence intervals. I think that everybody thought her or Zenobia's paper, but it's quite uncertain, but it's very old. And the genetic data of that sample is very poor. So here, the way I like to display is that I split up all the data that we have into either whether it has one of these stimulations or it does not. And then with the bars with the red bits are the things that we estimate to be contaminant, so actually from modern humans, and with blue is the bits that we estimate is actually from the Neanderthals. And you can see in this particular specimen, we estimate about 90% of the data is actually not from this Denisovan in this case. And you can see that it's for the amination, it's only about a third of the data that's contaminant, versus for the endogenous, it's almost all. But still, if you look at sort of the non-deaminated, it's almost all. But still, if you look at sort of the overall data, about 50% of the data that we have used is sort of using in this, doesn't have a deamination, right? So we do actually about double the amount of useful data that we have by using all the data and not just focusing on this data here. And this for specimens like this one here, it does make a difference, right? So because if you look at sort of what we get as the posterior decoding of this, you get something like this. So here, I think the colors are slightly different, but orange, we have Denisovan ancestry, blue, homozygous Neanderthal ancestry, and brown, you have heterozygous ancestry, right? So this is a Denisovan, so almost everything is orange. But you can see there's a few regions, for example, here or here or here, but this individual very clearly has quite long Neanderthal ancestry fragments. And so these fragments are, yeah, might seem like short bits here, but these are like, right, so these are in, this is in centimorgans here. So 1 centimorgan very roughly corresponds to 1 megabase, or roughly 1 million bases. So many of these fragments here are several million bases, so up to 10 to, I think, yeah, up to 10 million base pairs long. So these are big chunks of the genome, which sort of indicates that genome must have happened quite recently, right? And so we can then sort of ask, does this actually make sense, or is this just like a fluke? And so one thing we can sort of, instead of using this very fancy model, we can sort of look at a very simple thing. And here I look at just at this long region here, which is sort of the longest thing where we think we have some Neanderthal ancestry. And we can look at the only sites where humans have one allele, which is sort of the 0 allele, and either Neanderthal or Denisovan, but not both have the other allele, right? So here, every bar is one site, and blue means Neanderthal derived allele, and the other two guys don't. Denisovan's Africans don't, and orange means only Denisovan's have derived allele. And so the point is that if you had contamination, right, you would sort of get the 0 allele here. So whenever you get like the orange allele, we can be pretty sure that it must be actually coming from a Denisovan, because we don't see this allele in a Neanderthal, and we don't see it in present-day people. And likewise, if you see a blue bar here in our individual, it's very likely from a Neanderthal, because we don't see this allele in present-day people, and we don't see it in Denisovans, right? And if you look at our individual, this is pretty much what we see in the region here, that we see have about 50% of the stuff is blue, 50% of the sites are orange, which is what we would expect from a heterozygous admix fragment, right? And then if you look at this bit that's right here, which is outside the region, we only see orange fragments, as we again would expect from individual that's a Denisovan, right? So it seems to work well for this particular fragment. I'm going to do this for a second Denisovan, Denisovan 8. It's a little bit younger, it's a little bit higher data, and you can see that you get, in some sense, nicer regions. So we have this very long fragment here on chromosome 1, where we're quite sure that this individual has Neanderthal ancestry. We also have, because this is actually a male, so the X chromosome is haploid. So we get this one very long region on the X chromosome where it's you only have Neanderthal ancestry, because it's haploid. You can only have one ancestry, not two. And we can also look at Denisovan 3, and this is a little bit tentative, because there's also the ones that we use as a reference, and we see that there might be some regions here where we also have some Neanderthal ancestry, but likely much less. And so, so essentially what we find that in the early Denisovans, right, we find lots of Neanderthal ancestry, whereas in the late Denisovans, we find much less. And then we can also look at Neanderthal, right? Kind of flip the script and look at, for example, the Alpine Neanderthal, which is a high coverage Neanderthal from also Denisova cave, so from the same site. And here, now everything is blue, right? Because it's a Neanderthal, everything gets sort of this inferred at this Neanderthal ancestry, but we also find a few regions like here, and here, where we think that this individual has Denisovan ancestry, right? And these seem tiny maybe on your screen, but these are still like up to about 1 million base pairs long. So still fragments are long enough that we're quite confident that these actually are true signals and not just a statistical fluke. Right? But then if you look at sort of a later Neanderthal from the same region, or Neanderthals that we find in Europe, we don't see Denisovan ancestry anymore. Right? So again, this thing that sort of, we look at the earliest sample, we see sort of evidence of gene flow, but then it disappears, right? And so what we would think that maybe there have been some interactions before 110,000 years, but then this disappeared. And so how does it make sense, right? Because what you might naively assume, if you have gene flow, then it should stick in your population, right? Because if you have people that have both Neanderthal and Denisovan ancestry and they have offspring, then they should have the same ancestry. Right? There's no reason why Denisovan ancestry should disappear. And so what I think happened is that this region in Siberia was very likely sort of some kind of sink region, if you want, sort of I could call it a hybrid zone, but it's a lot of people move much more frequently there than people moved away from there, right? That Neanderthals perhaps were mostly in Europe, but occasionally made to Siberia, and Denisovans were maybe somewhere in East Asia or South Asia or Oceania, I don't know, but occasionally made it up to Siberia. But there were always not too many Neanderthals in that particular region, so they never really made it back. You know, what essentially happened in Siberia stayed in Siberia, right? And that's why when you have later Neanderthals that lived.
We always say that too when we go there.
Right, you say that too?
We always say that too. What happens in Siberia stays in Siberia.
Yes. Anyways, yes, and so that's sort of what my interpretation of this data is, and yeah, so then I have sort of a second story that sort of in some sense corroborates that, which we published just last month in Nature, and this is sort of focusing on one population of Neanderthals from this Trigirska cave in Siberia, and again this has been a big sort of. The previous paper I all worked on, a previous story I always from published data, but here we actually worked on new data, and this has been essentially done by a former postdoc of mine, Laurit Skoff, but there have been a lot of people involved, including Tom and Katerina, and Matthias and lots of people in Leipzig and elsewhere, and this work would obviously not be possible without all of these people involved. But so that. So the interesting thing is that for this Trigirska site here, right? So this is this sort of relatively, for Siberian at least, late Neanderthal site, where we were able to actually generate quite a lot of data from specimens that lived roughly at the same time. So what we did is we did capture data. So we only looked at sites where we knew were variable between Neanderthals, because most of the specimens had relatively little DNA preservation, right? And so, but what we were able to do with this data is to almost double the number of Neanderthals for which we have data from, right? So in gray are so here are the different Neanderthals that have nuclear data sequenced, and on the y-axis you have the cover rate, right? And so you can see that the gray bars are the ones that have been published previously, and the blue and orange bars are the new ones. And so we have 12 new individuals. 2 of them are from Oklanikov cave, for which we have very little data, and then the rest are from Trigirska cave, where we have more data, and so we have focused on analysis mostly on this one site, right? And so the first thing we did with this data is to ask, are these individuals, or are these specimens actually from different individuals, or are some of them from the same individuals? And in order to do that, we use a method called read, and it's the idea is conceptually quite simple. So essentially, we just look at the difference between any pair of individuals and sort of take the average between them. And the average we normalize to 1, right? And so if you have 100 individuals, you have a difference that's roughly 1. But then if you're related, because you're, say, father-daughter, for example, or mother-daughter, or so, then, right, so you share some chromosomes. So there will still be some chromosomes that, we're called identical by descent, but they will have essentially no differences to each other. So they will, these two individuals will look, if you look at these normalized distances, will look less or more similar to each other. And so you can predict based on the differences that we get what the relatedness is. So if you have, for example, 2 specimens or 2 bones that will come from the same individual, then you only expect half of the difference you accept between unrelated individuals. If they're first-degree relatives, so father, daughter, or siblings, you get about 3/4 of the difference, and then second degree is about 82% of the difference. And so we can, using that, we can sort of do that between all pairwise comparisons and then see where they fall. And we can see here that so these are all the specimens that we have, all the pairwise comparisons. And the number here highlights the specimen ID of the ones that are not unrelated, right? And so we have 3 specimens, Trigirska 13, 19, and 63, that we are pretty sure come from the same individuals, right? So we have the comparison here and over here and over here. And then we also have 2 specimens, 17 and 7, that we think are first-degree relatives, right? So they have, are closely related, right? And so one thing that allows us to do is sort of make this diagram here, where here all the small circle are the specimen numbers, and to sort of conceptualize that, we add letters, right? It's just, we were like, some journalists ask us if we gave them names, and it's like, no, we're not creative enough for that. So we just gave them letters from A to whatever, right? So what we can do is we say like, yeah, so these we believe are first-degree relatives, right? So we then also think that, then the question is, what kind of relation is this, right? So it could be siblings, or father-daughter, or mother-son, right? Because one is male and the other is female. And so the one thing that we then looked at is the mitochondria, right? And so we find that they actually have different mitochondria, right? And so this very likely seems to us that it cannot be mother-son, right? Because in that case, they would have the same mitochondria, because the son would have inherited the mitochondria from the mother. So it's very likely that one is the father and one is the other is his daughter, right? And so the other thing is, the reason why one is, it's a nice finding, but the reason that we actually think this is relevant is that this sort of hints that the individual must have lived at roughly the same time, right? Because one is the daughter of the other, so they must have lived within a year from each other. And this is relevant, right? Because the dating for these time periods is quite, has fairly high uncertainties, right? So if you say they've lived roughly at the same time at dates beyond radiocarbon dating, this means they might have lived within a few thousand years, right? And so if you think someone that lives today and someone that lives, a few thousand years ago, that makes a big difference if you talk about, say, social organization of these people, right? And so the fact that we have individuals that are sort of indicates that probably actually lived together, right? And then we also find second-degree relative pair between A and Triglyceride A and Triglyceride L, again, sort of indicates that they probably lived roughly at the same time. And then the third thing we find are what's called mitochondrial heteroplasmies. So these are variants in the mitochondria. And so you know that you have like a few hundred copies of each mitochondria, or mitochondria in each of your cell, and there's sometimes variants in them, and they do get inherited, right? But they tend to be very short-lived. So they tend to disappear within a few generations. And so the fact that we find shared heteroplasmies, again, sort of indicates that these individuals very likely roughly lived at the same time, right? And so then what we can do is we can sort of establish for these sets of individuals that they lived roughly at the same time. We can ask, are they more similar to each other than the overall set of the individuals? And we find that they're not. And so at least that seems to suggest that there's, no evidence they lived, over a prolonged period of time. So we assume that they were more or less one social group, and sort of we think that they can, be roughly sort of interpreted that we sort of can look at their sort of social organization, and it's sort of one unit and not multiple units that we smush together. So what do we learn from that, or why is this? What can we learn? And so one thing we can look at is sort of the proportion of the genome that's in homozygous bits. Because homozygous means both copy of your genome has the same, or you have no differences between the two alleles. And this happens if your parents are close related to each other, right? Which can happen because of inbreeding, or it can also happen if you live in a very small population, right? And if you had a lot of rare inbreeding, you would expect to find it just in this one individual, right? If you have, say, sibling mating, and sibling mating is rare, then we might just find one individual with this pattern. But what we find in our studies that pretty much every individual that we have the data to do that has about 20 to 30% of their genome in homozygous genomes, which is much more than what we find in all, say, modern human reference individuals, or any other reference data that we found, with exceptions of gorillas, right? So gorillas there's two subspecies that are critically endangered, so they also live in very small groups that are very isolated from each other, and there we see a very similar pattern, that they have a large part of the genome in homozygous tract. So it seems to sort of indicate that they must have lived in very, very small groups. Then the other thing that was striking that Lauritz found is that if you compare the Y chromosome diversity to the mitochondrial diversity, so Y chromosomes, right, are only paternally inherited, mitochondria maternally inherited. And so if there was no social structure, we would expect to have roughly the same diversity after we control for different size and mutation rates that we know these two have. But that's not what we find. What we find is that the Y chromosome diversity is about an order of magnitude lower than the mtDNA diversity in these guys, right? And so this here is the mean coalescence time, which again normalizes for mutation rate differences and for differences in the size of these genetic elements. And you can see that there's almost no Y chromosome diversity here. And again, if you look at sort of comparative data from a large number of human groups, we see this pattern is very unusual. But we do, interesting again, find this sort of in gorillas, and these 2 gorilla species that we have comparative data for. And yeah, so you can look at the ratio and sort of get the same pattern here. And then so what Laura did sort of did there's a few explanations for that. So we looked at, can it just be variation in population size or generation time? And in both cases, we found that he has a very hard time to fit simulations to these scenarios. One explanation could be that if you have only a very few males contribute most of the offspring. So if you had, like in this case, there's 3 out of 150 males generate almost all of the offspring, about 80% of the offspring, and then you fit it very well. But what you need would be a very large sort of local effective size, right? And so this is sort of in some sense not what we see from the homozygous tract. So we also think this is not a likely scenario. But then what we find is sort of the scenario that we sort of favor is that if you have between 60 and 100% of female migration rates. So if you have like various small groups, but it's mostly females that move between groups, whereas males tend to stay where they're born. In such a scenario, the best fitting scenario sort of suggests that groups have maybe a size of around 20 individuals, we get a pretty good fit. We can also, like if you combine these 2 scenarios, if you have mostly males staying and only a very few males contributing most of the offspring, this also works quite well. But it doesn't improve the fit, right? So we think at least female must have mirrored much more than males in this population. We don't really know whether males, how skewed the offspring distribution was. And so, to summarize, so what we think we learned about this Neanderthal population is that we have data from 12 Neanderthals that we very likely think that they were contemporaneous. And so we have this picture where we have these very small populations, but there must have been multiple populations, right? Because we think females must have been able to move at least between 2 different groups. Because the diversity that we see in the sort of sex bias markers is very, very heavily female bias, right? About an order of magnitude more diversity on the mitochondria than the Y chromosome. Yeah, and so in some sense, that's sort of, I think I started with saying, essentially one of the goals of my work is sort of, add more complexity to what we know about these guys. And so, right, so in some sense, you have sort of this contrast that on the one hand, I think Siberia has been at times a contact zone between Neanderthals and Denisovans, but it's also been quite isolated, right? So they both came there and had local gene flow, but it sort of stuck at this local scale. And there's not much evidence, I think, for gene flow out of Siberia to other parts of the world. And in summary, sort of, I think, this is I think one of the reasons why we need careful modeling, or what we can get from careful modeling and also novel data, because it allows us to really do genetic analysis over long time scales, right? You don't have to extrapolate from present data. We can really sort of look at how things evolve over time, and we get a much more complete and much more complex picture through that. And with that, we'd like to end. Mostly all the folks in Leipzig, my group, and also the ERC, who is currently funding my work. And you all for your attention.
Okay, thanks a lot. Questions? Microphone.
I've got loads of questions, but I'll try and just do one at the moment. Maybe we can talk later. So I was thinking about in the paper, one of the big questions is what you see in the Altai indicative of Neanderthal societies broadly? In other words, right across to the west of Europe. Mm-hmm. And prevailing thought now is, as I understand it, that based on high coverage genomes like Vindia, mm-hmm. that it isn't, that the Neanderthals are more, are less homozygous ridden, less inbred, as it were, in the West compared to the East. And therefore, that supports the idea, as you said in your talk, that you may have a situation where you get a smaller subgroup of Neanderthals sort of budding off and going to central Siberia and mm-hmm. to places like the Altai. Mm-hmm. So the question is it possible to say anything about how long that process might have taken? In other words, what do you see in the Altai? How long might that take? For example, could it be a very small group, a tiny group of Neanderthals of like 3 males and 10 females going there. And then 50 years later, or 100 years later, or 1,000 years later, this is what we see in the genomes that we can now detect in the Altai. Or is it not possible to say anything about that?
I mean, I think it's a little, yeah, I think it's a very good question. It's I think it would be hard, you know, to pin it down to any particular scenario, because I think there's there's multiple scenarios that would result in very similar genetic data, right? So I mean, so I think for the homozygosity to like you get to get this large amount of monocytes, I don't think you need very long time scales, right? So it could be that you have. I mean, sort of the same thing with the gorillas, right? We don't think they have had these very extreme population structure for too long. And it was like a big part, I think, is due to habitat loss. I mean, Martin, you actually correct me if I'm wrong, because I know you actually know these gorillas, but I think this is sort of, you know, that one of the reasons that this is because of that arising, and this happened, you know, in a
I mean, I guess we'll know with more genomes and more genomes. But
so I mean, I would think, you know, I think it's really hard to say anything. I think one can always speculate, right? But I think really knowing with, I don't think one can get a strong hypothesis.
Another sub-related question is about Denisovan ancestry in the Chagirskaya Neanderthals, of which there's virtually none, right?
Yes.
Does that surely tells us, perhaps, given that the localities of Denisova Cave v. Chagirskaya, I mean, these are very close places. Does that perhaps raise the possibility that when Neanderthals, like the Chagirskaya Neanderthals, are there, that there are no Denisovans at that point, and therefore tells us something about the fact that on occasion both groups were there, but sometimes there was only one group that was there.
I think that's possible again. But I think again, you know, we talk about such long time scales. You know, if you can be there in the same, you know, essentially, I think, you know, if you're there in the same century, right, it looks like they're almost at the same time, but you still won't meet. So I think, so there's a paper, I mean, I was pretty bad at updating slides, but there's been this awesome paper by Elena Zavala in particular, right, where she looked at sediments throughout the Denisova cave, and she has like a very high temporal resolution, so there's like 700 odd samples. And so she looks only at mitochondrial DNA. So this is, I mean, we're not so sure, but we find some Chagirskaya like mitochondrial DNA in almost the same layers, I think, as we find Denisovan DNA. So I don't, you know, from that data, one cannot make the conclusion that they have not been there at the same time. But again, you know, the timescales are so big, right? In some sense, it's. I mean, they're close, right? Yeah. I mean, it's not. Yeah. So I mean, I don't know if you, I mean, I can imagine if you follow some big animal or whatever, you know, that you end up in the same spot.
Together, it's pretty damn close.
Yeah. I mean, again, we talk about essentially, I think about lifetimes here, right? It's you probably don't meet every week, but do you? Yeah. I mean, I think it's. I mean, it's fun to speculate. Right? It's really. I mean, I think this is. Yeah. I mean, I think these are the things that are really hard to like get out from genetic data, right? So, I mean, I could imagine if one can show that they, like exchange artifacts or something, what might be able to get to the frequency of interactions. But I think very often it's also like,
Well, thank you very much. My question, in a way, I quite like I mean, the data is there, it's not whether I like it or not, about the Chagirskaya Neanderthals being a relatively well-knitted group and not necessarily having much to do with the Neanderthals at Denisova or other parts of the Altai. In a way, that was well known even archaeologically. You know, the archaeological signature is unique in the region. It's just so distinct.
Right. So you refer to the tools, right?
Exactly.
Yeah,
the lithic tools are, and I remember back, 10, 15 years ago when we first visited the site and we were shown the lithics by the colleagues there, they were saying, what do you think? And both Tom and I were like, wow, this is where they could be coming from La Ferracie or from anywhere in Western Europe and in France, for example, where you
right.
So these are very, very, very Neanderthal-like tools. Right. And but anyway, so that is what it is. But what is also very interesting is that even in the older individuals, like the Denisovan individuals, there is some Neanderthal integration.
Yes.
Which to us and to you guys suggests that these kind of interactions were not just, that 60,000 years ago, a small group of Neanderthals arrived at Chagirskaya and stayed there and whatever. We have this big interaction, the big long journeys happening, as you said, certainly until 110,000 years ago, so last interglacial. Mm-hmm. And do you think, and as more data comes out and looking at the Denisovan genomes, this will become the norm. This will be the norm when we see, or in terms of,
I mean, I would love. I mean, I think, the thing that would really help would be if you have the 7 genomes from different places.
Yeah.
Right. I think that would really. I mean, there is some people that I think have hypotheses that Denisovans are essentially Neanderthals or something Homo erectus-like, right? And if we could imagine something like this, I think that that what we just our models are all just completely wrong, and I think in Siberia, I mean the data suggests that it has been common, at least for the or at least to find essentially the 2 out of 2 early Denisovans I was able to look at, right, which is not a lot, but it sort of suggests that there is, that it has happened, right? But it's again, I think it's really hard to extrapolate from that. I think it's it's one of the challenges we're working at, as you know, right? That I think we need data from, say, China or some other place.
Okay. We have one question online. If others want to ask online, they can do that as well. So it's an easy one, I think. Dheeraj Chaudhary is asking, considering that our species were mixed with unidentified extinct hominin species in the past, can traces of extinct hominin species be modeled only with modern DNA if ancient DNA was not available? Hosted mixture, basically.
Yeah. So I mean, there's a lot of methods to do that, and so Martin has been one to evaluate those. It tends to be a lot harder, right? And I mean, there's a few things, right? So there is, people. I think there's at least a model that quite a lot of people believe that Denisovans have some ancestry from an unknown archaic population, but it also becomes sort of a very conceptual question at that point, because the modeling space is very large, and I think it's not always obvious that these people lived in very distinct groups genetically or socially. Where, for example, I mean, one case where people have studied as well is sort of early African modern humans, and so there people have been sort of making quite similar claims. There must have been some sort of some unknown populations, and then this is I think quite nice modeling paper by Simon Gravel from McGill, who essentially showed that it's really hard to distinguish sort of these sort of new lineages from just long persistent structure, right? That you might get if you have sort of a hunter-gatherer population that's just occupies a very wide area, right? Because if you have they have a species that also Neanderthals that say live in Siberia and Europe, right? They probably don't move back and forth all that often. And then depending on how often you observe them, you might think there are different species or different lineages, or you might just call them Neanderthals, right? I think it's really,
I think it's really the problem that, I mean, in Africa, we don't know a lot of things that we know better in Eurasia, right? Just from ancient genomes, that's something that we don't know as well.
Yeah, so I mean, I think it's really, really hard to make claims about unobserved things, right? It's sort of the same thing with sort of the question, do modern humans have Neanderthal ancestry? And I think this question was really settled from when we had Neanderthal genomes, right? Before that, you could find well-respected scholars argue either way. I think it's just, yeah, in turn, it's very hard to make observations of things that you don't directly observe, right? I mean, you can always get, evidence, and I think, it's don't get me wrong, it's still, I think, important that we do this kind of work, but I think it's also important that you acknowledge that this work will have limitations.
Okay. Questions?
Okay, thank you for the talk. It was very interesting. And in fact, actually yesterday, I had a guest, completely non-scientific person, and she told me, have you ever heard about this? And I said, yeah, actually, we have a speaker tomorrow. We're going to hear more. So I have two questions. One is that when you look at this local ancestry, as far as I understand, you look at basically the basically not the heterozygous but homozygous sites in Neanderthals and in Denisovan, and you have good high coverage, and then based on that you just say which part is from Neanderthal and which part is from Denisovan, right? So you have high coverage, but I didn't really understand that what's the length of the genome that is covered. I'm interested to know how many private SNPs did you look at in Neanderthals and the Denisovans, and is that proportional to the length of the chromosome? And basically, because the thing is, like, I'm wondering, like, yeah, what's the number of the SNP basically that you look at this local ancestry?
Yeah, so I think there's two parts, right? So first, so for this method, actually, I don't only look at the homozygous SNPs, but I look at all the SNPs for which we have data, right? So I have, I mean, to give the technical answer, right? So this is this here in some sense is the likelihood here, and so the A and D here are the number of ancestral derived alleles you observe at each site for a particular ancestry, and so we look at about 3 million SNPs, right? And so. But if you're correct that most of the information actually, or a lot of the information comes from the ones where all the Neanderthals have one allele and all the Denisovans have the other allele, right? But I don't know the exact number of those that have this particular pattern, which will have the most data, but the others also do contribute to the likelihood.
And then is it possible, like in future, basically using your data, like set up a pipeline and just said basically if you have very low coverage data, if it's based on just homozygotes and we have really low coverage data, just basically you plug it in and either is this or that. Because you don't have, you're not, if you don't look at the heterozygote site, you just basically can still find out how much ancestry, like Neanderthal ancestry or Denisovan ancestry you have in that particular sample that has very, very low coverage. If you only look at the private alleles.
Wait, so are you, I'm not quite sure I understand. So the question is, can you just ask whether it's a Neanderthal or Denisovan? Is that
kind of, yeah.
Yeah, so you can do that too. I mean, you can also do that, yeah.
Plug it in quickly and say like basically either has this or that.
Yeah, I mean, you don't need most of the stuff that I do here to do that, right? So there's sort of similar methods that people right? I mean, you might have heard, for example, of this D statistics that people use, right? So if you remove if you remove sort of all the sort of stuff along the genome bit, you get something that's very close to, say, one of these D statistics, right? Where it just will tell you over the entire genome, you look more like a Neanderthal, you look more like a Denisovan.
And the other question, the second one that I have is this 20 to 30 person inbreeding that you found out in the samples just from this site. And you said that it's just basically very similar to the other species that they have been extinct or close to extinction. I'm wondering, like, is it just based on the percentage of the inbreeding, or did you also have a chance to look at the mutation load and basically figure out like which part of the genome it is, and if it has a negative in the
yeah, that's a good question. So we have not been able to do that, and so I think, I mean, it sort of glossed over because I realized I ran a bit short on time, but so we actually don't have full sequencing data for the Cherkasy Neanderthal. We only have capture, and we captured only about a few 100,000 sites that we knew were variable between different Neanderthals and Denisovans, right? But then we want to look at the load, right? You want to look at, ideally, you want to look at rare variants where ideally you know that they are, you know, that they've messed up some protein or whatever, right? And for those, we don't really have the data, so we haven't really looked into that.
Thank you so much.
Yeah.
You have one more? One more.
One more.
Sorry. Okay, so I'm really, it's really exciting about the whole recombination and when did admixture happen using recombination and stuff like that. Sorry. And when you look at, for example, Ustashim or Oase, you see there are some big chunks of Neanderthal integration, integros DNA, but there are also lots of little bits as well. So there's an argument that it could be that there's been two phases of admixture, two pulses of admixture, or it could be potentially that there isn't. There's maybe one. So my question is about looking at these chunks of DNA and working out, you know, A, how long ago did that event happen, which I understand the basics of, which you explained quite well. But B, is there a kind of a way in which you can sort of assess whether or not it's likely that there's one admixture event, even though we've got different sizes of these chunks? I mean, how do you make a determination of whether it's two or one?
Yeah, so one of the things that we do, and I mean, it's something that I'm currently working on formalizing, but the one thing you actually can do is not look at the length, but you can look at the location of the fragments, right? Because if you had sort of the same, because you know that overall you have not all that much, in this case, Neanderthal ancestry in Denisovans, right, or Neanderthal ancestry in humans. So it's a few percent, and what is very likely, you know, that essentially, in some sense, the first few generations depend which part of the genome will actually will have Neanderthal variants in. For example, so one thing that I'm currently doing is to look at the Neanderthal ancestry throughout modern humans, and just ask, you know, through time in modern humans, ask how is it correlated between different individuals. And so what you find there is that you have a remarkable, like the pattern you see is extremely similar if you just look at the overall genetic diversity, right? And so to me, this says it's very likely just genetic drift. You know, Neanderthal admixed in some early human groups, and then it's just been genetic drift. The exception of that is are individuals like Oase or Bachokiro, where we find long fragments. And so what I think is, if just looking at the sort of correlation of location of fragments, is a very good sort of proxy to say, you know, what was the source of the ancestry. And so if you do that here between the two Denisovans, so this Denisova 2 and Denisova 8, which is here, we actually find that the location of fragments are uncorrelated to each other. So I don't think that they must have been the same event. I mean, the other thing is looking at the, like, if you, I mean, one thing is that the dating of Denisova 2 is quite vague, right? So it's in principle possible that they might have lived at the same time, even though I think they come from very different places of the Denisova cave. But if you sort of take the sort of median date at face value, right, then you would not expect, you know, if you sort of think that the event happened shortly after, say, 167 years ago, you would expect the fragments in the later Denisovans to be much shorter. And they're not, right? Sort of at least occurs in this at this time period, there must have been recurrent gene flow. So I think because the dating is a little bit uncertain, I would say the location of fragments are the stronger arguments, but the dating, I think, adds to that as well.
Okay, I also see that Rebecca Rex-Sykes raised her hand. I think she can just unmute herself and ask a question, right, Rebecca?
Hello, hi there. Shall I put my video on? Hello, I really enjoyed the talk, of course, and it's not really a question, but something that I found really exciting about this, if I've interpreted your data correctly, is that you have 3 fragments from one individual. So you have 13, 63, and 19, which are all from individual G. at the site. And one of those pieces, 19, is a naturally exfoliated deciduous tooth. So what I found interesting from that, from the perspective of how we understand that Neanderthals are using their landscape and their presence and the frequency with which they're at sites, that seems to suggest that you have an individual who was there, lost their tooth naturally, and then later they've died there. So is that's what the data is saying, right? So
I wish. I mean, okay. I mean, I have to say first, I'm not an expert on that. I'm just quoting Ben Saviola here, who did that analysis, right? And so.
Oh, this is from your supplementary materials in the description of.
Exactly. I mean, Ben is the co-author on this paper, right? And so he did that part of the analysis. So I can only quote him. So it's part of what we published, right? And so I believe it's to be correct. And we had the same idea, right? But Benze, I mean, could not exclude a scenario where, you know, the tooth was essentially lost during this individual dying. So we can put, because I think if you sort of look at the age estimate, right, I think one of the tooths based on where looks like it belonged to a slightly older individual. And at first we were very hopeful, you know, that we could exactly show what you described, the scenario that we sort of, that these people must have come back to the site. But yeah, Benze thinks this is not the only scenario.
Yeah, I mean, that's what would be nice to know, isn't it? Because that's the thing that.
No, I agree.
Yeah. How often are they actually using their sites?
They just all, exactly. Yeah. I mean, it would be fantastic, but yeah, unfortunately, we cannot. Yeah.
All right, that was all I had to say. Thank you very much.
I would have to ask Benze. I can just because
all
again,
right, thank you.
I don't have the expertise for that.
Thank you.
Okay, cool. Then I think that was a very nice discussion as well.
Yeah, thanks for.
Okay, one more.
This picture more in detail. What is this big chunk of Neanderthal segment in Denisovan 8? It's like, what is it? What is so big? And there's like all Denisovan chromosome X.
Yeah.
And then the big track is Neanderthal.
Yeah. So this is because I think I mentioned this very briefly. So this is because it is a male. So it only has one X chromosome.
Okay.
Right. So we don't have heterozygous ancestry on the X chromosome.
Okay.
We only have either Neanderthal or Denisovan ancestry.
Okay.
So that's the reason why it is. I color it blue because it is just one ancestry.
Yes.
Yeah.
Okay. Thanks.
Okay, good. Then